Hello everyone, this is Vaibhav Shandelia, MBBS student from ESIC Government Medical College, Hyderabad, and today we're going to talk about a very important disease which is Filariasis, and this is mainly caused because of Wuchereria Bancrofti, which accounts for 90%, and also rarely this is caused by Brugia malai and Brugia timori, but we won't discuss that right now. Mainly we'll focus on Wuchereria Bancrofti. So we'll study this under the life cycle, pathogenesis, clinical manifestation and lab diagnosis, which is required for the examinations. And let's get started. So starting with the life cycle, the vector for this disease is Culex quincke fasciatus. Okay, Culex quincke fasciatus, but it can also rarely be transmitted by Anopheles and Aedes, but mainly remember Culex quincke fasciatus. So now this mosquito bites and transfers the infective form, which is the L3 filariform larva. Okay, so the infective form L3 filariform larva has been transferred into the human, and it gets access to the local lymph nodes where it molts twice, okay, and develops into the adult. There the adult fertilizes to form microfilariae in the blood, and then sheathing occurs. Sheathing is basically formation of an envelope like structure around the microfilariae, and then the mosquito bites again, and this L1 microfilariae is transferred into the mosquito. This is where X sheathing occurs. Basically the microfilariae molts twice and then forms the L3 microfilariae again, and this migrates to the proboscis of the mosquito, which is the part that stings the human, and then again it bites the human and then transfers the L3 filariform larva again. So this is the life cycle. Basically L3 filariform larva to adult to L1 microfilaria in the blood to L3, and then it bites back. So this is the main life cycle. Next we go to the pathogenesis. So we have to know that pathogenesis can occur because of the adult worm and the dead worm both. And so due to the live adult worm, we see that there is lymphatic dilatation and thickening of vessels. Okay, so basically this has a lot to do with the lymphatic system. That is where most of the pathology lies. So adult worm will cause lymphatic dilatation and thickening of vessels, while the dead worm will release toxins and metabolites which will be harmful for our body. Together these antigens will trigger a host immune response, and here there occurs infiltration of plasma cells, eosinophils and macrophages. At the same time, the lymphatic system becomes tortuous and it also gets damaged. So damaged lymphatic vessels, tortuous lymphatics and infiltrations. Okay, this is the host immune response in response to the antigens by the adult worm and the dead worm released by them. Also, remember that this sort of an symbiotic relationship going on, where till the worm is alive, the lymphatic system is damaged of our body, but it still works. It will still serve its purpose, but it's damaged. But as soon as the worm dies, there occurs a granulomatous reaction because of the antigen secreted by the worm, which causes fibrosis of the lymphatics and causes lymphatic obstruction. Okay, remember this. And just a quick factoid that endosymbiotic relationship is seen amongst the Wuchereria Bancrofti and Wolbachia of Rickettsia. Now coming to what happens next is the host immune response. Now going deeper into this mechanism, the antigen presenting cells present the filarial antigen to the macrophages, where the T helper cells then differentiate into Th1 and Th2 subsets. Okay, now Th1 Th2 subsets will, in early infection, both of these are activated, while in a microfilaric individual in which a microfilaria has infected an individual, Th2 is mainly stimulated only, while Th1 is downregulated. And this increase in Th2 actually causes hyper IgE syndrome, increases IgG4, which is specific to the parasite, causes eosinophilia, and there is also an increase in IL-4, 5, 10, 13. If you've read immunology, you'll remember some of these pathways. So also in chronic filariasis, Th2 is increased again, and here also IL-4, 5, 10, 13 presence is seen, and also parasite specific IgG1, 2 and 3 increase. So basically what we have to note here is that the antigen presenting cells bring the filarial antigen and help in conversion of the T helper cells to Th1 and Th2 specific. And this Th1 and Th2 are basically, the Th2 is stimulated, which causes hyper IgE syndrome, IgG4 increase, eosinophilia, and IL4, 5, 10, 13 increase, mainly in acute filariasis. While in chronic filariasis, IgG1, 2 and 3 are also increased. Okay, and remember the adult worm and dead worm causing different pathogenesis. Next, coming to the clinical manifestations, this is divided into 4 stages. Okay, which one are they? They are basically the manifestations are lymphatic filariasis, and this is divided into 4 stages: endemic normal, asymptomatic microfilaria, acute filariasis, and chronic filariasis. Okay, so let's start with endemic normal. This means that normal people in endemic area which are not infected. So these are just normal people, but they are in a very endemic region. Okay, next we have asymptomatic microfilaria, which basically means there are no symptoms shown by the affected individual, but the filaria are still present in their body. Okay, so basically they can manifest as hematuria or proteinuria, or there can be tortuous lymphatics, or under USG we can observe the filarial dance sign, but main symptoms have not yet appeared. Then comes the stage of acute filariasis. Here there is high grade fever, lymphadenitis, lymphangitis. The lymph node become tender, firm and enlarged, especially the femoral, inguinal, axillary and epitrochlear lymph nodes. Okay. Then the male genital lymphatics show epididymitis and orchitis. Okay, these are affected as well. There is early pitting edema, which can be resolved by raising the leg, because that the gravity helps to dislodge the edema. Then there is dermatolymphangiosis, which is lesion on affected skin with inflammation. Okay, coming to chronic filariasis, this is seen 10 to 15 years after the infection, and here granuloma forms, and this causes pedal edema and causes the disease elephantiasis, as you can see in this image, and also other lymphatic obstruction can lead to hydrocele. At the same time, chyluria can be observed, which basically means milky white urine. Okay, so this was lymphatic filariasis and its 4 stages: endemic normal, asymptomatic microfilaria. Here USG filarial dance sign can be observed. Acute filariasis, where there is high grade fever, lymph nodes enlarge, lymphadenitis, lymphangitis, orchitis, epididymitis, dermatolymphangiosis is there. Okay. Then comes chronic filariasis, where we see granuloma formation. This occurs 10 to 15 years later. Chyluria occurs. Okay, now there are other manifestations as well apart from lymphatic filariasis, which are tropical pulmonary eosinophilia and manifestations because of the immune complex. Coming to tropical pulmonary eosinophilia, this is occult filariasis, which basically means hidden. Occult means hidden. So here the microfilaria are present not in the blood or the lymphatics where they usually are, but instead they are dislodged in the lung. Okay, in the lungs, and hence they cause an allergic response. Basically this is hypersensitivity reaction to the microfilarial antigen. Okay, because the microfilaria are lodged in the lung. This is tropical pulmonary eosinophilia. Next we talk about the immune complex manifestations, which this immune complex can be deposited in the kidneys or the joints. And in the kidneys it can cause nephrotic syndrome, hematuria and proteinuria, while in the joints it can cause arthritis. Okay, so we saw manifestations, we saw lymphatic filariasis, tropical pulmonary eosinophilia and immune complex manifestations, right? Now coming to the lab diagnosis, the specimen taken for lab diagnosis is the blood, hydrocele fluid or urine, and under the direct wet mount we can observe classical serpentine movement. Thick thin smear, Leishman or Giemsa staining is used, and what we observe is a sheath and nuclei of the microfilaria. Okay, now concentration techniques are used to better detect the microfilaria or filaria in the blood, and this is to increase the sensitivity of tests, and this is membrane filtration and not centrifugation. These are 2 concentration techniques. Now coming to the collection time, when samples should be collected, this is a very critical step because the microfilaria are not always present in the blood. If it is nocturnal filariasis, then the microfilaria will come out from 9 p.m. to 4 a.m. in the night. But if you want to collect the sample at the daytime, we have the DEC, diethyl carbamazine provocation test. Here DEC tablet is given orally and the microfilaria come out automatically in the blood within 15 minutes to 1 hour. Okay. Now coming to the quantitative Buffy coat examination, centrifugation of blood and then staining with acridine orange, we can under fluorescent microscopy we can observe the microfilaria. Okay. Now antigen detection, OG4C3 antigen and AD12 antigens are present, and this can be detected by ELISA or rapid immunochromatography testing. Antibody detection, IgG4, anti-sheath antibody are present. Flow-through assay shows Wuchereria Bancrofti SXP1 antigen, which is the antibody to WP123 antigen. This is the luciferase system. This is something very specific, but if you want to remember, you can remember that there is WB-SXP1 antigen and WB123 antigen, okay, which are detected using flow-through assay. So this is the antibody detection, mainly IgG, IgG4 and anti-sheath antibody. Okay, you can remember these because this is present in the pathogenesis. This is increased in the pathogenesis, all three of these. Okay, now imaging methods used. Ultrasound can be used to observe tortuous vessels, serpentine movement and filarial dance sign. Okay, remember filarial dance sign. Then X-ray can show detect dead calcified worms. Okay, so this was the lab diagnosis. Okay, let's get a quick overview because this is a very important organism. So life cycle, because of Curex quinquefasciatus. Okay, there is transfer of L3 filariform larvae in the human. Then lymph nodes, molt twice to adult, fertilized to form microfilaria, sheathing occurs. Then transferred back if the mosquito bites again, and then molt twice to form the L3 microfilaria, and then goes in the proboscis of the mosquito to infect human again. Pathogenesis. Due to live adult worm, there is lymphatic dilatation and thickening. Due to dead worm, there is release of toxins, and host immune response will cause infiltration of different plasma cell eosinophil macrophages and tortuous lymphatics and damaged lymphatics. There can also be secondary bacterial fungal infections. Then till the worm is alive, nothing will happen to the lymphatics. I mean, it will be damaged, but they will still work. While after the worm dies, due to granulomatous reaction and fibrosis, obstruction will be seen. The host immune response, we see that antigen presenting cells cause conversion of the T helper cells into the Th1 and Th2 subsets, which then the Th2 increase and Th1 down regulation causes eosinophilia, IgG4 increase, hyper IgE syndrome, and IL-4, 5, 10, 13 increase. Then we saw the clinical manifestation on lymphatic filariasis divided into 4 stages: endemic, normal, asymptomatic microfilaria, acute filariasis, and chronic filariasis. We saw tropical pulmonary eosinophilia and immune complex manifestations, right? Lab diagnosis, and mainly what we have to remember here is that we see the sheath and nuclei of microfilaria under Leishman or Giemsa staining. Okay, DEC provocation test is used for collection if you want to collect samples in daytime. Okay, then antigen OG4C3 and AD12 are present, and at the same time IgG, IgG4 and anti-sheath antibodies are present. Okay, and definitely ultrasound shows tortuous vessel, serpentine movement and filarial dance sign. This is the main features of Wuchereria Bancrofti. Now something we have to know as well for examinations are the difference between Wuchereria Bancrofti and Brugia malai. This is a very famously asked question. You'll get this sometime in the viva mostly. So here what we see that there is presence of in the head end, there is a head end and there's a tail end, and this is where the feature differ between the both. So at the head end, sheath is present in both. Okay, but remember, you see this cephalic space. Cephalic means head. So this empty space in the head area, in Wuchereria Bancrofti, this is present in 1:1 ratio. Okay, while in the Brugia malai, the cephalic space is 2:1. The cephalic space is increased. Okay, and also the next main feature you have to know is that at the tail end, this is the most important thing, there is no nuclei in the tail tip in Wuchereria Bancrofti. There is no nuclei. Okay, while in Brugia malai, there are 2 widely spaced round nuclei in the tail tip. Okay, 2 widely spaced round nuclei in the tail tip. These are terminal nuclei. Terminal nuclei are not present in Wuchereria Bancrofti. This is the main differentiation. Okay, so this was the complete video on Wuchereria Bancrofti, which causes filariasis. I hope you liked the video. Thank you.